All right, folks, welcome back to the lymphatic system. So now we're starting to get to some of the meat of the lymphatic system. The next couple mini lectures will be the majority of where your studying will take place. All right, so before we get going into what the cells do and how they do it, let's talk about how they communicate with each other. The communication between these cells happens through chemicals we call cytokines, and we name some subpopulations of cytokines. For example, some of them are called chemokines. We call some of them interleukins. Interleukins are cytokines, interleukins. We call some of them interferons. Whoops, can't spell. That's supposed to be a T. There's the F, interferons. So we have all these cytokines, and they're different different subcategories of these cytokines, and they're named by what they do and where they go. Interleukins is probably a huge family of them. Interferons, we'll talk about how they do antiviral things. Alpha and beta interferons do antiviral. We'll talk about that. The gamma just makes macrophages better macrophages. So we'll talk about these. This is probably the most important classification of cytokines, and that's your interleukins. And by the way, there's lots of them. There's interleukin 1, 2, 3, 12. I don't even know how many at this point. There's lots and lots of interleukins. Interleukin 1 and interleukin 2. We'll talk a lot about. Interleukin 1 is involved in fever production, so we'll talk about that. All right, so cytokines, interleukins, for example. Interferons. Thing I want to tell you about interferons are the alpha and beta interferon put our cells into an antiviral state. Gamma interferon enhances macrophagocytic activity, so it enhances the activity of macrophages. We have some other chemicals like tumor necrosis factors. Of course, they do what they sound like they're doing. They stop tumors from growing, but they have some other activities as well. It's just that the first thing we saw them do was stop tumor growth. Matter of fact, I'll tell you a story really quickly. There's a guy down in New York City named Dr. Coley. And this is back in the early 1900s, by the way. And Dr. Coley was an oncologist. And he noticed that one of his patients went into remission. And he didn't understand why. And as it turned out, there was some salmonella infections going around. Now, back in this day, salmonella was bad. Matter of fact, a lady became famous down there, a food worker. Her name was, I forget her last name, but her first name was Mary. And of course, that's Typhoid Mary I'm talking about. So salmonella was a pretty deadly disease back in that day. I mean, it still can be deadly, by the way. And Dr. Coley noticed that one of his patients went into remission, and there was a salmonella infection going around. And he wanted to understand why his patient that was infected with salmonella would have tumor would go away. And as it turns out, tumor necrosis factor is a protein secreted during inflammation and infection. And it also does what it sounds like. It causes tumors to die. Tumor necrosis. Necrosis means death. So in this case, it was the infection with a pretty bad gram-negative bacterium, gram-negative rod called salmonella, that led to high tumor necrosis factor that led to remission of the cancer. Now, how cool a story is that? That's a pretty cool story. So we have these other chemicals that help with our immune system here. Not only interferons and interleukins, but tumor necrosis factors and some other cytokines. This is the collage showing you all of them. Well, it's not showing you all of them. This is really just a smattering of cytokines. In fact, I have my office mate has a poster in our office of all these cytokines. It's a pretty impressive poster. And I don't even think that poster is all inclusive. There are just way too many of these to sit there and memorize them. But now you know they exist. So now you know that in your future life you could look these up if you had to. All right, so let's talk about our innate immune system, the immune system we're born with. That's what we're talking about right here, innate immunity. There's 7 major categories within our innate immune system. First of all, there's the physical barrier. That's the skin and mucous membrane. Physical barriers, skin, and mucous membrane. There's the generic cells of the immune system. And when I say generic, it means they don't respond against any one particular pathogen. They respond against all pathogens pretty much equally. And these cells are called phagocytes. And sometimes we say macrophages because that's the major class of phagocytes, but it's not the only. Macrophages come from monocytes. All right, but we have other phagocytic cells like neutrophils are phagocytic. So you might hear people toss around the word macrophage interchangeably, but there are other phagocytic cells. We have natural killer cells. That's another cell that we are born with. That it surveys our blood and body fluids and kills things. And then we have a bunch of chemicals. We have interferons that we already talked about. The alpha and beta do antiviral states, and the gamma enhances phagocytosis. We have a series of complement proteins, and these complement proteins put holes in the pathogen and kill it. And there's over 20 of these complement proteins, and we'll talk about them a little bit. And then we have an inflammatory response. The biggest thing about inflammation is it's chemotactic. And what that means is you attract white blood cells to the area. That's what chemotaxis means. Chemotaxis means you secrete a chemical that attracts attracts something to the area. In this case, it would be attracting a white blood cell to the area. And fevers, what they do is they take the bacteria out of its optimal temperature growth, out of its optimal temperature range for growth, so it's suboptimal temperature for growth for the bacteria. And it also increases our phagocytic activity. Our cells are bare phagocytes when our body gets warmer. So we'll talk about fever a little bit and how it helps us. Now I say how it helps us. As long as the fever is not too high for too long, it helps us. But of course, the proteins in our brain won't withstand a fever that's too high for too long. And you could get brain damage from it. So you do have to treat a fever if it's too high or lasting too long. But a transient fever, a transient mild fever would help you fight off infection. All right, this is showing you the physical barriers, skin and mucous membrane. And yeah, these are things you should know. The physical barrier, skin and mucous membrane. Some of these secretions are antimicrobial, but we'll talk about them when we talk about the chemicals. So these secretions, like your sebum in your sweat, reduce bacterial growth. But right now we're just saying this is a physical barrier. We'll talk about the chemical later. We have a series of cells that fight off infection pretty generically. That means they'll phagocytosize everything about equally. They're not honed in on one particular pathogen. We have natural killer cells, which do immune surveillance. It's a pretty important word because that's how we all say it. When you see immune surveillance, you should think natural killer cells. And then let's start the chemicals. We have different chemicals that help fight off infections, like interferons. Please remember that alpha and beta interferon are antiviral. All right, that's interferons. And then I'll add some more chemicals on this slide. Some of our secretions have things like defensins. What a cool thing to name a chemical that fights off infection, right? Defensins. There's lysozymes in our secretions. Lysozymes. Lysozyme has a pretty cool little story to it. Here's what happened. Sir Alexander Fleming, a British scientist, he accidentally sneezed on one of his microbiology plates, and he noticed that around his nasal secretion the bacteria died, and he was working that up, studying it, and he discovered lysozyme. Lysozymes in our tears, our sweat, our saliva. So lysozyme is an enzyme in our secretions, and it kills bacteria. And he discovered this by accident because he sneezed on a plate growing bacteria. And while he's working up lysozyme, he left a loaf of bread on the counter, which grew some mold on it. And the mold invaded one of his plates that had bacteria on it. And around the mold, the bacteria died. And then he discovered penicillin. So how lucky could a man get that he sneezes on a plate and discovers lysozyme, and then accidentally discovers penicillin while he's working up lysozyme. So that's a pretty cool story. And a reporter asked him, Sir Alexander Fleming, aren't you a little embarrassed that both of these major discoveries were quite by accident? And he said no. He said chance favors the prepared mind. He coined that phrase, which is used a lot now, but he coined it. Chance favors the prepared mind. Okay, there's two classes of phagocytes. There's microphages, which are your neutrophils, and eosinophils. These are called microphages. And then there's macrophages, which come from your monocytes. And when the monocytes leave the bloodstream and set up shop in a tissue, they're then called a fixed macrophage. And we have lots of fixed macrophages like dendritic cells in our skin, Kupffer cells in our liver. So these are fixed macrophages. Alveolar macrophages in our lungs. So we have all of these fixed macrophages. So we got microphages and macrophages. This is a picture of a natural killer cell, but what I want to point out is how it kills. What it does is it binds to this abnormal cell, and then it secretes a series of proteins called perforins. These are all called perforins. And the perforins make a hole in the membrane of the abnormal cell. And that allows solutes to exit and water to enter, and it lyses the cell. And you can see the lysis of the abnormal cell right there. Now, putting a hole in a membrane of a pathogen or an abnormal cell is not exclusive to just natural killers. Complement proteins actually do that, as you'll see shortly. So this is a recurring theme, putting holes in the membranes of abnormal cells or pathogens. Natural killers happen to do it with proteins called perforins. Perforins perforate. Think that. Perforins perforate. Again, interferon alpha and beta. I should have organized this better because I keep repeating myself, but alpha and beta cause our cells to go into an antiviral state. Our cells make antiviral proteins, and I'll show you how that happens in a second. Interferon gamma doesn't do antiviral state. What it does is it increases phagocytic activity. It enhances our macrophages. This is how interferon alpha and beta work. The first cell right here to be infected by the virus is doomed. It's pretty much shot. It's going to die. But it secretes interferon. And here's the interferon right here. It secretes interferon. That interferon, alpha or beta, goes to a neighboring cell, and in fact all of the neighboring cells, and it turns on some genes that make antiviral proteins. Now, when this virus bursts out of this first cell and tries to infect this cell, these antiviral proteins digest the viral nucleic acid, so it can't even get a foothold. So yeah, the first cell infected is doomed to die, but it saves all its neighbors by secreting interferons before it dies. And that's how interferon works.
When a cell is infected by a virus, the virus enters the cell and produces structures that are not found in uninfected cells. The presence of this viral material signals the cell to produce interferon. The interferon moves out of the cell and attaches to receptors on nearby cells of the same type. The cell that produces the interferon is unable to save itself. The virus replicates in this cell and then moves out to infect nearby cells. The nearby cell that already has interferon bound to its surface responds in several ways, including production of enzymes that degrade messenger RNA and prevent protein synthesis. Thus, a virus can attach and enter the cell, but completion of the viral replication cycle is prevented.
There you go. That's how interferon alpha and beta work. Okay, complement. Complement is a series of proteins, there's over 20 of them, that will make holes. Well, they'll do two things. One thing is they opsonize, and what opsonize means is just coat for destruction. That's supposed to be an I, folks. You know my hand. By now you know my handwriting is atrocious. Opsonization is just coating for destruction. It makes our phagocytes better at gobbling it up if something's opsonized. So complement can just coat a bacteria or coat a pathogen, and that's called opsonization. In addition, complement can actually put holes in the membrane of a pathogen and kill it. So it can kill it by a membrane attack complex. That's what the hole is called. It's called a membrane attack complex. Or it can just opsonize and wait for a phagocyte to come along and gobble it up. So it does it both ways. That's what complement does for us. Another defense mechanism is inflammation. And what inflammation basically is leaky capillaries. All of the signs and symptoms of inflammation, redness, swelling, heat, pain, can be attributed to three chemicals: prostaglandins, histamines, and kinins. And prostaglandins, histamines, and kinins make your capillaries leaky. And this is showing you the histamine story, because mast cells secrete histamine. So with inflammation, we cause our mast cells to degranulate and secrete histamine. Blood flow increases. Phagocytes are activated and attracted. Chemotaxis occurs. Capillary permeability increases and blood leaks into the area. Now listen, blood leaks into the area, and that's why your area is red, because blood is red. Blood leaks into the area, and that's why your area is warm or heat, because blood is warm. Blood leaks into the area, and that's why your area swells, because you just added a bunch of blood to the area. It's swollen. And in addition, prostaglandins, and I'll spell that correctly for you, although it's probably coming up here shortly. Prostaglandins cause pain. So let me spell prostaglandin for you. So this is inflammation. And as long as inflammation is not too extensive, where it causes further damage, it's a good thing. Acute inflammation is a good thing. Chronic inflammation can be very damaging, but acute inflammation can help you heal. So as long as the inflammation is not too extensive or chronic, it can be a good thing to help you heal. And lastly, we have fever here. Interleukin 1 is involved in the fever. Here's how it works. Interleukin 1 goes to your brain and causes your hypothalamus to secrete prostaglandins. Prostaglandins reset your thermostat. They say to you, 37.2 is no longer normal, 38.5 is normal. Now, if your brain thinks that 38.5 is normal, but you're at 37, you to yourself feel cold. And you start to shiver and try to heat up to 35. And you hit 37.5, and you hit 38, and you hit 38.1 and 38.2, and you're getting warmer and warmer and warmer, but you still feel cold. But when someone puts their hand on your forehead, to them, you feel feverish or warm, even though to yourself you feel cold. Now, as soon as you hit your target, 38.5, now you don't feel cold anymore because you've hit your target. And you feel feverish to the people that touch your forehead. Now you fight off the pathogen, and interleukin 1 goes away and prostaglandin drops. Your thermostat resets to 37.2. Now you feel hot to yourself because your temperature is 38.5, and you start to sweat. But when someone touches your forehead, you feel cold and clammy to them because you're sweating and all that sweat is evaporating because your temperature is high. So even though you feel hot and you're trying to cool down by sweating, you feel cold and clammy to the person touching you. So that's how a fever cycle works, with increasing your temperature and then having your temperature drop. All right, complement. 3 complement cascades. There's the classical cascade, there's the alternative cascade, and there's the mannose binding lectin cascade. But here's the cool thing. They all have this common pathway at the end. So really the only thing different in the complement cascades, the different pathways, when I say cascade, I mean pathway. Cascade can be used interchangeably with pathway, is how it starts. The classical cascade always starts with an antibody. The alternative one starts with a protein called properdin. And the mannose binding lectin always starts with mannose and lectin. And they all ultimately end up with this common pathway right here, which always does 3 things. Helps with inflammation, makes the membrane attack complex or cytolysis, and opsonizes a pathogen. So no matter which cascade you're in, you're going to lead to inflammation, cytolysis, and opsonization. This is the properdin pathway. P is properdin. Yes, it has two other proteins called B and D, but it goes to this common pathway, as you can see, and you get inflammation, you get the membrane attack complex, and you get opsonization. And lastly, I have the mannose binding lectin pathway. Mannose sticks off of our pathogen. Here's a mannose. We make lectin. Lectin binds the mannose, and this initiates the mannose binding lectin pathway. We call it the MBL pathway, mannose binding lectin. Looks like our book calls it just the lectin pathway. And this ultimately leads to the common pathway, which leads to inflammation, the membrane attack complex, and opsonization. So now your question is this: do I have to know all these intermediates in here? The answer is no. Know how the three pathways start and know how they end: inflammation, cytolysis, and opsonization. So those are the three complement pathways. This is showing you the classical pathway here because it starts with antibodies. It's going to lead to a hole in the membrane. This is showing you the alternative pathway here. It's not actually showing you the, it's actually not showing you P. So I'll just leave that alone for a second. So here you have the classical pathway right there going on, and it leads to a membrane attack complex, a hole in the membrane, which leads to cytolysis. Okay, so that's complement in a nutshell. All right. Now this is how complement leads to inflammation. C3a and C5a cause mast cells to degranulate. And remember, inflammation is due to prostaglandins, histamines, and kinins. Well, here's histamine right there. Here's your membrane attack complex, which is cytolysis. And C5b and C3b opsonize. It's not actually showing you C5b doing it, but it does. So C3a and C5a, histamine, C3b and C5b opsonize. They coat the bacteria for destruction. When something is coated for destruction, phagocytes can do their job better. So that's complement in a nutshell. This is showing you C3a and C5a degranulating a mast cell, and that's part of the that's just part of the inflammation pathway. Okay, so that's complement in a nutshell. You can see two different pathways feeding into the common pathway. The one that's missing here is the mannose binding lectin.
Complement consists of a group of serum proteins that activates inflammation, destroys cells, and participates in opsonization. Complement can be activated by a number of different foreign molecules. The complement proteins respond in a sequential manner, producing a cascade of reactions. The major components are C1 through C9, named in the order that they were discovered, not in the order in which they function. The complement cascade can be activated by the classical pathway or by the alternative pathway. In the classical pathway, C1 becomes activated when it binds to an antigen-antibody complex. The activated C1 then cleaves C2 into C2a and C2b, and C4 into C4a and C4b. C2b and C4b combine to form a protease called C3 convertase. C3 convertase then cleaves C3 into C3a and C3b. In the alternative pathway, antigens such as endotoxin, polysaccharides, or cell wall components react with C3b. Small amounts of C3a and C3b are constantly being formed from C3, but without activation they are soon destroyed. C3b reacts with the proteins factor B, factor D, and properdin to form a complex called C3 convertase, which cleaves C3 into C3a and C3b. Both of these pathways of complement activation follow the same sequence after cleavage of C3. C3a is involved in stimulating inflammation. C3b reacts with other complement components to form C3 convertase, which forms more C3a and C3b. C3b also attaches to the surfaces of microorganisms. Phagocytes have a binding site for C3b. Therefore, microorganisms with C3b bound to their surfaces are more susceptible to phagocytosis. Coating of bacteria to make them more susceptible to phagocytosis is called opsonization. Addition of properidin to C3 convertase results in formation of C5 convertase, which cleaves C5 into C5a and C5b. C5a enhances inflammation and acts as a chemoattractant for phagocytes. C5b reacts with other complement components including C6, C7, C8, and C9 to form a membrane attack complex. This structure forms a hole in the cell membrane and causes cells to lyse.
Okay, so that's how complement works. 3 different initiation pathways, and they all end in the same common pathway that makes membrane attack complexes, inflammation, and opsonization. All right, so this is how we talk about inflammation. There's the signs and symptoms of inflammation right here. Heat, redness, swelling, pain, and sometimes loss of function. This is when you want to start maybe treating inflammation, when you start having loss of function. But inflammation is not necessarily bad as long as it's not too extensive. And we say, by the way, here are some Latin words for the swelling is tumor, the redness is rubor, the heat is calor, and the pain is dolor. That's just the Latin base for these words. So this is inflammation, heat, redness, swelling, and pain. All can be accounted for by the chemicals prostaglandins, histamines, and kinins. Prostaglandins, histamines, and kinins. Here are the effects of inflammation. First of all, it hastens repair. Secondly, it seals off the pathogens from the rest of your body. It acts like a barrier. It isolates them. It isolates the pathogens. That's important. You don't want these pathogens getting into your bloodstream. So it limits the spread of the pathogens, in other words. And it keeps the infection local instead of systemic. Meanwhile, we can mobilize our defenses even though they're systemic defenses, but we keep the infection local. All right, what are the products of inflammation? Well, when tissue dies, that's necrosis. The tissue will die, you'll have necrosis, and our body will have to reabsorb all that dead tissue. The white blood cells might die, and if the white blood cells die, you get pus. I mean, they're going to fight off the infection, but they could die doing it, and that's the source of pus. And if you get an accumulation of necrotic tissue and pus, it can lead to an abscess. Sometimes you have to intervene in large abscesses and lance them and lyse them and treat them with some kind of topical agent if it was a skin abscess, say. But abscesses sometimes can go away on their own as well as your body fights off the infection. So this is going to step you through the process. Here's a sliver or something going into your skin, and the mast cells are going to degranulate and release histamine. Because the mast cells degranulated and release histamine, you start the inflammatory process. You have chemotaxis. Chemotaxis is attracting other white blood cells to the area. All right, you have white blood cells coming to the area. They fight it off, and then you repair. You have a granuloma and then repair going on. So this is just inflammation and repair in a nutshell. That's the collage showing you inflammation and repair. The big thing is inflammation leads to chemotaxis and activation of these phagocytes.
The inflammatory response is an important nonspecific defense against tissue damage. It begins when injured tissue cells release chemical signals that activate the endothelial cells of nearby capillaries. Within the capillaries, adhesion molecules called selectins are displayed on the activated endothelial cells. These adhesion molecules attract neutrophils, slow them down, and cause the neutrophils to roll along the endothelium. As the neutrophils roll along the endothelium, they encounter chemicals that activate integrins, which are adhesion receptors on their surfaces. These integrins then tightly attach to adhesion receptor molecules on the endothelial cells. This causes the neutrophils to stick to the endothelium and stop rolling. This accumulation of neutrophils along the walls of the capillary is referred to as margination. The inflammatory mediators released by the injured tissue bring about changes in the environment that cause mast cells to degranulate and release histamine. Histamine causes vasodilation and an opening of the junctions between the endothelial cells, allowing fluid and leukocytes to leave the capillary and enter the infected tissue. The neutrophils now undergo dramatic changes in shape and squeeze through the endothelial wall into the interstitial tissue fluid. This process is called extravasation. The neutrophils, followed by other types of phagocytes, are attracted to the damaged site by chemotactic substances released by bacteria and tissue breakdown products. They ingest and destroy invading bacteria.
Okay, folks. So extravasation, by the way, has another name. Another name for extravasation is diapedesis. Diapedesis. So the video just called it extravasation, and that's fine. Extravas is vessel. Extravasation, outside the vessel, crawls outside the vessel. Diapedesis is another word that means the same thing. So you need to know both words, extravasation and diapedesis. All right, fever. Fever takes the bacteria out of its optimum temperature range. Here's how it works. First of all, we engulf a bacteria. We secrete interleukin-1, goes into the bloodstream. That tells our hypothalamus to make prostaglandins. Prostaglandins reset our thermostat. So now instead of being 37.2, it's 38.5, say, something like that. And our body heats up to 38.5 because that's what our thermostat is now set at, and that's the fever. Now, these things that reset our thermostat are called pyrogens, and there's some endogenous pyrogens, that's interleukin-1, and we have some chemicals from bacteria that are exogenous pyrogens that can cause fever in us that we didn't make. So we have pyrogens that can be both exogenous and endogenous. And this is a collage showing everything. We've talked about every single one of these now. Okay, folks, see you next section.